Hey folks, Arush here, and today we're talking about deoxyribonucleic acid, or just DNA for short. So let's go and talk about this. We've already spoken about Gregor Mendel and his contribution to genetics, and now we're going to talk about these three individuals. We have Rosalind Franklin and another team of scientists called Watson and Crick. And in the 1950s, scientists knew that DNA was responsible for carrying genetic information from parent to children, parent to offspring. But what they didn't understand is they didn't know how it worked. Now, before you could figure out how something works, you have to find its structure. If you could identify something's structure, then you're pretty sure you could figure out how it works. So in the 1950s, you have this race to find the structure of DNA, and you have scientists everywhere, searching to find the structure. And we happen to have this team of scientists by Watson and Crick, and we also have this young lady, Rosalind Franklin. And they're independently working on discovering the structure, trying to be the ones that find the structure of DNA. And what ends up happening is over time, these three individuals end up meeting, and during their meeting, they come across this image. They see this picture that Rosalind Franklin took. Now, Rosalind Franklin, what she did was she used X-ray diffraction, which is where she would take molecules and place them on like a crystal, and then shoot X-rays at them. And those X-rays would give certain images. And this happened to be an X-ray diffraction image that she got of DNA. Well, Watson and Crick, they knew quite a bit of information about what DNA was composed of, but they just couldn't figure out the structure. Once they saw this image of the crystallography of DNA, they were able to figure out that DNA was it's a double helix, or it looks like a twisted ladder. Now, later on, after a few weeks of seeing this image, Watson and Crick were able to find DNA structure probably about a few weeks after seeing that image, and they were awarded the Nobel Prize, whereas Rosalind Franklin, since she worked with X-rays, she ended up passing of cancer shortly after. So if we continue to look at DNA, we could see that it looks like a twisted ladder or a double helix. What that means is there's two strands, and instead of running perfect with each other, they're actually twisted around. The word DNA, deoxyribonucleic acid, this portion of the word, deoxyribo, is actually coming from this portion, deoxyribose. Oops, gotta fix that. Deoxyribose. And we know that anything that ends in -ose, anything that ends in that suffix, we know that's a sugar. So go ahead and keep that in mind as we talk about the structure of DNA. So we learned earlier in this year that DNA is a polymer, and it's made up of smaller building blocks, and we call those building blocks nucleotides. Okay? If we look at a nucleotide, there's three components, three major parts that make up one nucleotide, or one building block of DNA. So this circular part, we have a phosphate, we have the sugar, and specifically, this sugar is deoxyribose, okay? And then the third part is we have a nitrogenous base. All three of those together, they form one building block or one monomer of DNA, one little piece of DNA. If we look at the nucleotides, this phosphate and this sugar portion is always going to stay the same. However, this nitrogenous base, there's 4 different variations, or 4 different nitrogenous bases that we could find in DNA. Go ahead and add this to your notebooks as well. DNA is a polymer made up of the monomer nucleotides. Make sure you highlight that too, please. So the 4 nucleotides, we have, number 1, we have adenine. Number 2, we have thymine. We have guanine, and we have cytosine. If you notice, if you were to look at the structure of adenine and the structure of thymine, you would notice that those two match each other, kind of like puzzle pieces. And we call that, when two things complete each other, we call that complementary. So we would call adenine and thymine complementary bases because those two bases fit perfectly together. Okay. Notice that the phosphate and the sugar, though, that portion doesn't change, only the nitrogenous base. That's the only variation. If we look at guanine and cytosine, again, we can see that guanine and cytosine complete each other. They're complementary bases. Okay. An easy way to remember how or which nucleotides complement each other is this little mnemonic. We have apples and trees, or adenine pairs with thymine, and cars and garages, okay, or cytosine pairs with guanine, and vice versa, okay. If we look at these different monomers, or these different nucleotides, you can see that it starts to build this larger structure. Now, one thing about DNA that's interesting, and we're going to talk more about during DNA replication, is if you know the nucleotide sequence on one end, you can predict what the nucleotides are going to be on the opposite end. You would just know that by their complementary base. So if I have a thymine right here on the opposing strand, I would have an adenine. Okay. If I have a cytosine here on the opposing side, I would have a guanine. If I had a guanine, the opposing strand would have a cytosine. And finally, if I had an adenine here on the opposing strand, I would have a thymine. Another important part about DNA is the backbone. Now, when we're referring to the backbone, we're specifically referring to these two molecules, the sugars and the phosphates. Those two molecules make the backbone, or give DNA its structure, okay? So if I were to give you an image like this and ask you to label it, it would go phosphate, sugar, phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, okay? So the two molecules, even though there's three molecules that make up the nucleotide, okay, the three molecules that make the nucleotide is the phosphate, sugar, and the base. These two are what we're talking about when we refer to the backbone of DNA. Okay. Another thing that you'll notice about DNA is the two strands run anti-parallel. What that means is they're running in opposite directions. So if I go back to these, you'll notice that in order for this nucleotide to match on the opposing strand, it has to face the other direction, okay? And we call that antiparallel, okay? One strand is going this way, the opposing strand is going the other way. So, why is the sequence of DNA important? How does that relate to living things? Well, if you guys have seen The Matrix, you've seen the opening scene, which has binary code. What binary code is this sequence of ones and zeros, ones and zeros, ones and zeros, and that's all it is, just ones and zeros. But that combination of ones and zeros determines something about a computer program. It could be a word or how a computer program is going to work. And all that information is stored in this sequence of ones and zeros. Okay? Well, just like those ones and zeros control a computer program, what makes us unique and different is the combination of A's, T's, C's, and G's. It's the nucleotide sequence. Okay? So what makes you and I different from each other is this. We have a different sequence of nucleotides. We have a different sequence in our DNA, and that's why we're different. So that was DNA structure, and I hope that was helpful.